Hello and welcome back to Beaver Science. Today's graphic is going to be related to Unit 2 and eukaryotic cell structures. Remember, there are three major anatomical structures in eukaryotic cells, those being the plasma membrane, which we see outlined here in figure 5.2, and that's essentially what we're going to do our drawing of today. The other two major subanatomical structures in the eukaryotic cell consist of the cytoplasm and the organelles contained within the cytoplasm. Remember, the job of the plasma membrane primarily is to control what comes into and out of the cell. And so we're going to draw up this phospholipid bilayer and the various globular proteins that are kind of randomly arranged throughout that phospholipid bilayer. Let's get this out of the way, and you will need the first sheet of study guide worksheet number 1 for the drawing. So I'll give you a minute to track that down and we'll get started. This is going to be a fairly busy drawing, and so I'm not sure I can keep the entire drawing basically in the field of view as we go. And we're actually going to use pretty much the whole top half of the page, all the way from the left to the right hand side. Now, since this is the first time you'll be doing this drawing, you may want to turn just a blank sheet of paper or your study guide page to the back side and do this in landscape instead of a portrait. But anyway, here we go. Let's give it a try. You need to start out by basically just drawing the phospholipids. And so we're going to start over here on the left-hand margin, pretty close to the left-hand margin, just about an inch down from the text above. And the drawing's not too difficult in terms of the structures. It's basically a series of circles and lines and maybe some oblong shapes that represent the globular proteins. But just start out by essentially drawing a series of 2 to 3 of these phospholipids. And remember that the plasma membrane consists of the phospholipid bilayer, and so this is one of the layers of the plasma membrane, and this is the other layer of the phospholipid bilayer. And then kind of randomly arranged throughout this phospholipid bilayer are the globular proteins. And a globular protein obviously is a protein. It's made up of amino acids, but a globular protein has a little bit more complex structure than a simple protein within the primary structure, which would just be a linear chain of amino acids. And we'll look at these globular proteins and talk about basically their functions as we draw up the membrane. So I'm going to rotate the paper here for just a minute, and we're going to label this the phospholipid bilayer. And what the phospholipid bilayer does essentially is it separates the two solutions that we find in living organisms. One of those solutions is inside the cell, and then the other solution is everything outside the cell. Basically, everything that's outside the cell we're going to call extracellular fluid. And that extracellular fluid could be blood, it could be water, it could be interstitial fluid. But essentially anything that's outside of the plasma membrane is going to be extracellular fluid. All the fluid in the interior of the cell we just refer to as cytoplasm. And so the phospholipid bilayer, the plasma membrane, separates the extracellular fluid from the cytoplasm, and its major function basically is to control what comes into and out of the cell. And so we'll be talking quite a little bit about molecular transport and also the way that molecules basically cross this barrier. And we say that the plasma membrane is semi-permeable. that means that some molecules can move freely across this phospholipid bilayer and some molecules can't. So those molecules that are small and nonpolar, things like oxygen, carbon dioxide, they can move directly across this phospholipid bilayer. So if we've got a whole bunch of oxygen out here outside of the cell, and we don't have very much oxygen inside in the cytoplasm, I hope my pen holds up. Those molecules will move basically following the concentration gradient. So those molecules will move from an area of higher concentration to a lower concentration, seeking equilibrium. Here we have a higher concentration of oxygen in the outside of the cell in the extracellular fluid than inside of the cell in the cytoplasm. So this oxygen will tend to move and follow down the concentration gradient and go from a higher concentration to a lower concentration, seeking equilibrium. That's fortunate for us because we need that oxygen to do cell respiration to generate energy in the form of ATP. That reaction of breaking down glucose in the presence of oxygen happens in the mitochondria. And the byproduct from that process, from cell respiration, is what we breathe out, and that's CO2. So we get an accumulation of CO2, higher concentrations of carbon dioxide inside the cell in the cytoplasm than there is outside of the cell in the extracellular fluid. And because CO2 again is a small nonpolar molecule, it can freely move back and forth across that phospholipid bilayer. And so it's going to move from an area of higher concentration to an area of lower concentration, seeking equilibrium. When molecules move based on this concentration gradient from a higher concentration to a lower concentration, seeking equilibrium, that process is called diffusion. And I don't think that's a terribly difficult concept to understand, but as we go further in and talk a little bit more about molecular transport, we'll take a look at what the consequences are for the cell based on these movements back and forth across the plasma membrane in terms of volume and pressure inside the cell. So let's continue on just a little bit here and add a couple of more of these phospholipids. And one of the things about the phospholipid, hopefully you'll notice that there's a head region and a tail region. Okay, the head region of the phospholipid is what they call hydrophilic. And philic means loving, and so hydro is water. So this portion of the phospholipid molecule is hydrophilic. It's basically a phosphate group, and so it will attract water. And that water basically will set right on the surface of the membrane, both out here in the extracellular fluid and also up against the inside of the membrane on this side of the cytoplasm. That gives fluidity to the membrane. And so the phospholipids and the globular proteins can actually slide back and forth within the membrane. So that's that phospholipid bilayer. The head portion is hydrophilic, water-loving. The tail portion is hydrophobic. And so these are lipid tails. And if you think about lipids, think about oil. And you know that oil and water don't get along very well. So the tail region of this phospholipid molecule basically repels water. And that prevents a lot of water basically from moving back and forth across the plasma membrane between the lipids, the phospholipid molecules. But water will move, and we'll take a look at how the majority of the water moves back and forth across the cell here in just a minute. When water moves, it'll move through one of these globular proteins that we've been talking about. Okay, there's two major groups of globular proteins. The first major group is a group of proteins called peripheral globular proteins. And if you hear peripheral, that kind of means that you're on the outside looking in. And so peripheral globular proteins aren't embedded through both layers of the phospholipid bilayer. They're just kind of attached to the surface or one side of the phospholipid bilayer. And so those are called peripheral globular proteins. And there's just one group or category of the peripheral globular proteins, and they're found on both sides of the phospholipid bilayer. So we could draw them out here as well if we had a little bit more room. But they could be attached to either the extracellular fluid side or the cytoplasm side. They provide structure and support basically for both sides of the phospholipid bilayer. So let's just add a couple of more phospholipids in here. We're going to do a total of 6 of these globular proteins. And so this thing's going to get pretty busy by the time we get over here to the other side of the page. So again, if this is the first time you're doing it, you may want to do it on a separate sheet of paper or on the back of a piece of typing paper that you could attach to your study guide worksheet too. Okay, the next sequence of proteins are what we call integral proteins. And integral proteins penetrate both layers of the phospholipid bilayer. And so these integral kind of means integrated. And so these globular proteins basically are integrated through both layers of the phospholipid bilayer. This first one that we're looking at, the identifying characteristic or trait of this globular protein is this unique carbohydrate chain that attaches to the outside of the globular protein. Sometimes they'll call these glycoproteins. And so this is a carbohydrate chain. And this is unique to each individual cells. And so this is our carbohydrate chain. The function of this particular globular protein, it's called the cell recognition globular protein, and each one of these proteins is kind of named based on its particular function. And so if you think cell recognition, well, it has something to do with identifying the cell. And so this particular protein identifies the cell as belonging to the individual. It's part of your immune response system. And so this is called the cell recognition. A lot to write here, and again, this is a busy drawing. Hopefully we can get it all in on this one page, and but you have to be really judicious with your use of space to get this all in here. So the cell recognition globular protein identifies the cell as belonging to the individual. It's part of your immune response system. And so if you have foreign cells or like a virus, your cells will be able to recognize that as being kind of an invader. And essentially it can identify that cell as something that needs to be taken care of by your immune system, by your white blood cells. Sometimes that can create a problem for a person who maybe is undergoing a organ or tissue transplant. And when they talk about, rejection of an organ or tissue, it may be because those carbohydrate chains aren't recognized or are seen as a foreign cell, and your body can actually attack those organs and those tissues. The next globular protein, again, all the ones that we're going to look at from here on out are integral globular proteins. And so they're going to be through both layers of the phospholipid bilayer. And this one has this little unique binding site, and it's kind of like a little lock and key mechanism where certain types of molecules will attach to turn on chemical reactions inside the cell. And so basically hormones and enzymes will interact with this binding site. And so say this would be a hormone or an enzyme, and it has a specific kind of fit that will interact with that binding site and turn on chemical reactions inside the cell. So some of you may know people who have trouble, balancing their glucose levels because they don't produce insulin, or the insulin isn't recognized by the cell. And so this would be an example of like a binding site for insulin, or maybe like adrenaline. And what that does when that hormone or enzyme attaches to that binding site, it turns on or sends a chemical signal to the cell to perform a specific function. Take up glucose, start to make more ATP for energy. And so this particular globular protein, we'll have to move over here a little ways to label it, is called the receptor globular protein. And so the receptor globular protein basically interacts with hormones and enzymes to turn on or promote chemical reactions within the cell. The series of the next three globular proteins are all involved in molecular transport. And so I'm going to just go ahead and kind of draw those up and see if we can't get everything on one page here. And then I'll come back and talk a little bit more specifically about the function of each of those globular proteins. So, first thing is, let's get a little bit of room maybe for our labels. And so we're going to add a couple more of these phospholipids. And then we're going to draw in these other three globular proteins. The first one is just a channel, or it kind of forms a conduit between the extracellular fluid and the cytoplasm. And then let's just kind of keep on trucking here with our phospholipids. And then we have one that kind of looks a little bit more like a Pac-Man shape. And again, we'll try to squeeze one more in here at the end. And these last two are both called carrier globular proteins, but they have a little bit different function. This first one kind of looks like a channel globular protein, but I tend to draw it a little bit more rounded just so you could distinguish between the channel and the first carrier globular protein. The last globular protein has these binding sites associated with it, and there are actually three on one side. On the other side, there are two binding sites. And so instead of a letter E, it looks like that maybe smiling face that you carve out of a jack-o'-lantern on Halloween. And then just so we know that this phospholipid bilayer continues on, let's see if we can squeeze a couple of these on the end. Yep. Alrighty, so we've got our globular proteins drawn in now. Let's back up and label these and talk a little bit about the functions of each of the globular proteins. This one, or all three of these, are involved in molecular transport. And again, remember, the major function of the plasma membrane is to control what comes into and out of the cell. So the first one that's involved in molecular transport is called the channel globular protein. And the channel globular protein basically just kind of forms this conduit for molecules to move back and forth across the membrane. And an example of this, we talked about diffusion a little bit, but there is a specific type of diffusion that occurs when we're talking about water molecules that's called osmosis. And there's a type of channel globular protein called an aquaporin that allows water molecules to move back and forth across the plasma membrane. And so this little Mickey Mouse looking thing basically is our H2O. And so hydrogens, two hydrogens and an oxygen molecule there to represent the water. And when water moves through this process of osmosis, it follows the concentration gradient. So again, it's going to move from a higher concentration to a lower concentration, seeking equilibrium. So if we have a whole bunch of water out here in the extracellular fluid and not much water inside of the cell in the cytoplasm, that water is going to follow the concentration gradient. And it's going to go from a higher concentration to a lower concentration, seeking equilibrium. Now, as long as all the molecules can move back and forth freely, you don't really develop a problem with volume or pressure within the cell. But that's not always the case. Some molecules can't move, and you know, if you see plant cells sometimes that either have not enough water or too much water, that can create issues for the cell and animal cells as well in terms of volume and pressure inside of the cell. So that's the channel globular protein. The next one is the first carrier globular protein, and so we're going to do two of these carrier globular proteins, this being the first one. So this one is carrier globular protein, and this is number 1. Both of these, the channel globular protein and the carrier globular protein, they don't invest energy to move molecules back and forth across the membrane. It's a passive process just based on the concentration gradient. And so I'm going to turn the page here for just a minute and write the term in here, passive. And that means basically that there's no energy being invested by the cell to move these molecules. Okay. This first carrier globular protein will actually change shape and kind of help to move certain types of molecules basically across the membrane, but it's still going to follow the concentration gradient. An example of a molecule that kind of moves this way is that 6-carbon sugar that we're familiar with, and that is glucose. And so this particular protein will actually undergo a shape change to help facilitate, it's called facilitated diffusion, and help facilitate the movement of certain molecules across the membrane, in this case glucose. It's still going to follow the concentration gradient. And so let's write glucose in here just so you know that that's the molecule that we're talking about. But it's going to go from a higher concentration to a lower concentration, seeking equilibrium. No energy investment by the cell. I'm going to back up here in just a minute and add H2O over here. So that reminds us that we're talking about the movement of water. And that movement of water is a specific type of diffusion called osmosis. Okay, both of these proteins do something called facilitated diffusion. And so it's like it's facilitating, it's helping to move these molecules basically back and forth across the plasma membrane. So this is so it's still diffusion, but they're getting a little help along the way from their friends here, these globular proteins. The channel globular protein and carrier globular protein 1. This last one is carrier globular protein 2. And so let's go ahead and label that. And this one moves those molecules that are either too large or are polar. And if you hear that term polar, basically that means that they have a charge associated with them. And so a lot of times these are ions. And one of the really important kind of ion exchanges that occurs through these carrier globular proteins is what they call the sodium-potassium pump. And this carrier globular protein actually does use energy. And I'm going to use these little dots basically to represent the sodium. And the thing that's different about this is this particular globular protein will use energy to kind of buck the concentration gradient. So it will actually move molecules from a lower concentration to a higher concentration using energy. So for example, here we don't have very many sodium ions inside the cell on the cytoplasm side, but we might have more or a higher concentration of sodium ions outside the cell. You would tend to think, well, those molecules are going to move from a higher concentration to a lower concentration seeking equilibrium. And that would be true if we were talking about a passive process of diffusion or facilitated diffusion or osmosis. But here we're talking about an active type of transport. So this is called active transport. And the cell is actually going to invest some energy to move these molecules. I don't know if I can squeeze anything out, any more life out of my red pen, but I'm going to give it a try here. And so we're going to look at two different types of ions, basically a sodium ion and a potassium ion. So we'll try this one. And so we'll use basically a triangle to represent the potassium ion. And so outside in the extracellular fluid, we have a lower concentration of the potassium ions, and inside the cell, we have a higher concentration of potassium. And this is part of what they call that sodium-potassium pump. And so that's that ion there. And we'll go ahead and label the sodium out here as well. So under this scenario, the cell's actually going to invest energy. Again, I'm going to turn this for just a second. And the energy that the cell uses to pump these molecules against the concentration gradient is ATP, adenosine triphosphate, our old buddy that is kind of the ultimate energy source for chemical reactions in all living things. So what happens here is basically, I'll draw one more sodium ion in here. That sodium will attach to one side of this carrier globular protein using energy in the form of ATP. And the potassium will attach to the other side, and the cell will use energy basically to move those molecules against the concentration gradient. So lower concentration of sodium inside the cell, it's going to actually move from a lower concentration to a higher concentration, kind of bucking or going against that concentration gradient. And the potassium is going to move from a lower concentration to a higher concentration inside the cell, again kind of going against the concentration gradient. And what happens is you end up building a kind of this large, what they call an electrochemical gradient, and you're storing energy across the membrane. And so that stored energy can be used basically to drive chemical reactions. And so there are proteins that will allow that sodium and potassium basically to rush back and forth across the membrane, and it generates like a little electrical response. And that's part of how your nerve cells fire to basically, stimulate a response in your muscle cells or other cells in your body. Okay, this process is not passive. This process is called active transport. And again, it occurs across that carrier globular protein number 2. And one of the things that I didn't draw up for you, but just to be aware of and remember that we talked about, I'll kind of sneak over here and see if I can find a little spot on the page to do it. but is that phospholipid molecule. And so I want to add that just real quickly here to kind of finish up before we wrap up this drawing. And so the phospholipid, basically, remember you have a phosphate head and then you have the lipid tails. Okay, this portion of the molecule loves water, and so it is what they call hydrophilic. And I know this drawing's gotten busy and a lot going on. Philic means loving, hydro is water, so that's going to attract water. And then the tail portions are lipids, and so lipids are kind of like oils, and so those are hydrophobic. And again, what that does is it helps to ensure that you have water on both sides of the membrane, but you don't have a lot of water moving back and forth across the phospholipids. And the water's going to move, but the water's going to move based on a concentration gradient, and it's going to go through these channel globular proteins, specifically the aquaporins. So yeah, a whole lot going on there. Let me see if I can find just real quickly here a finished graphic that you'll be able to download from Canvas. And so I'm going to trade this out with a drawing that I did earlier that's kind of a little bit more complete, and maybe take a minute to get that centered, and hopefully you can see all those things and use that to help you as you complete this portion of the study guide. Alrighty, thanks for joining. Hope you'll come back and see us again.